So just wanted to welcome everybody to today's ABC's Training Tuesdays, Part 7. We have David Haviland from UC Extension at Kern County to talk about mites and ways you can manage them. Just wanted to remind folks that there are ways to get DPR credits and CCA credits, and Ashley and Miranda, once they are able, will tell you how to do that. And just by the way, I'm Gabrielle Ludwig. I'm Director for Sustainability and Environmental Affairs at the Almond Board of California. I'm stepping in for Drew Walter, who's off becoming a daddy for the first time.
Miranda. Yeah, welcome to the webinar this morning. This is an interesting experience as we all learn how to do these kinds of things remotely. And the invitation I got today was to talk about spider mites and their management. And it's kind of a, this is a topic I've been working on for 20 years, and it's kind of a fun time in my career in that, typically how it works as a researcher is you write a proposal, you do the work, you write a report. You write a proposal, you do the work, you write a report. And you do this every year, year after year. Well, over the last year and a half or so, we've had some chances to do some what I might call culmination type of work. But also with COVID and a sabbatical leave I was able to take, I was able to kind of change that. So instead of doing, 1 year, 1 year, 1 year, 1 year, it was time to take a look at 20 years worth of work and try to pull it all together into something that's meaningful. And so we've gotten a couple of good peer-reviewed publications. Third one actually found out this morning is they're recommending a revision on it, but that's the way things work. So that all this should be able to get published, which as a scientist is good. But of course, as a scientist, frankly, I could care less about those publications. It's that the information gets in your hands that's what's most important. So that's kind of what today's about. So some of today will be a little bit of background. I realize we've got people that are probably new to mites and people that are probably 30+ year veterans. So I'll do a little bit of the basic, but also a little bit of the what you do. And hopefully along the way, explain why you do what you do. I want to teach principles, not just give steps. So just to start out, basic biology of spider mites. So on the top left up there, that's what you'll see during the winter, or in reality, you probably won't see during the winter. That is what we call a diapausing female. So in the winter, all of the mites are females. They have this orange color you see in the top left picture. And they're tucked down in nooks and crannies, in the bark and in places that they can hide all winter. About the middle of March down in Kern County up to 1st of April, as you move farther up, those mites will come out and they'll move up into the crotches of the trees and they'll start feeding on the leaves. Spider mites, specifically, I should say Pacific spider mite. There are other species, but Pacific spider mite and two-spotted spider mite, they feed exclusively on leaves, not on anything else whatsoever. And they can reproduce very quickly. So in the summer, in a week, for example, they can do an entire life cycle from egg through nymph, larval nymph stages into an adult. And the other thing about spider mites is they're associated with webbing. So that's where the name spider, one of the reasons they get the name spider mites. Now, what's the damage that they cause? So spider mites, they actually have stylets, kind of like little needles for mouth parts. And they use that, they thrust these needles forward and they poke individual cells in the surface of the leaf. And as the cell gets punctured, a little liquid comes out and they drink that up. So each time that happens, you get a dead cell that turns into a little mark that we call a stipple. You can kind of see on the picture here, sort of those little yellowy spots on the leaves. So you'll get 1 or 2 of those, then 10, then 20, and so on until the point that all those little stipple marks start to coalesce. You get this browning necrotic spots. Eventually, the tree recognizes that this leaf just isn't pulling its weight anymore. Photosynthesis it's not doing its job. And when that's the case, time to abort. And the leaves will senesce and fall off the tree. So obvious defoliation. Well, we know that trees need leaves. They need photosynthesis. So anytime that you start losing leaves, you're at risk of decreased photosynthesis, which of course affects carbohydrates. Now, you'll note my comment here on the bottom right nevertheless, almond trees are very resilient. And I can't highlight that enough. Imagine you lose a few leaves in the top of the tree. Well, okay, the sun hits the next leaf or the next leaf or the next leaf. I hope that nobody walks in an orchard and says, I've got a few browning leaves, I've lost a few leaves, this is a nightmare, because that's not true. There's a few studies out there, not a lot, but a few studies on the impacts of defoliation. Most of those studies are in baby trees, 5, 6, 7-year-old trees. And definitely on a 5, 6, 7-year-old tree, if you lose all the leaves, you're going to stunt the growth of that tree and you're going to have a pretty good impact on yield. At the same time, once a tree gets older and established, 10 years or beyond, you can actually lose a lot of leaves and not have any impact on the fruit at all. And when you do have an impact, it's not the current year, it's in the next year. So the sort of nightmare scenario would be you lose a whole bunch of leaves right before harvest or right during harvest, and then the tree uses all of its carbohydrates to push new leaves out when it should be doing bud differentiation. If that's the case, you can get a bunch of vegetative buds instead of reproductive buds, which then affects the number of flowers the next year and potentially the number of fruit. So as I said, if you get a yield loss from the mites, it's not in the current year, it's in the next year. But, thinking about it, if you go from, let's just pick a number. If you have 1,000, if you should have had 1,000 flowers per tree, I'm just making up numbers here, and you end up with only 800, but in all reality, the tree can only carry 500 nuts. If you lose 10% of your buds, from fruiting to vegetative buds, but you still have way more buds than the number of nuts the tree can carry. At the end of the day, you're going to come out similar. Likewise, if you have a few less nuts, you'd end up getting bigger nuts instead of a few more nuts that are smaller. So, all these things interplay, and it's really complicated to calculate. So that's why, nobody can give you a definitive number on how much defoliation you could sustain. So, I can't do that, but I can just say, trees can take a lot. So, take that into account. Oh, there we go. So just a close-up picture. I know this isn't an almond leaf, it's a plum leaf here, but just to show you what I'm talking about, you can see little stipple marks. Initially, you can see sort of bronzing as all those pull together. You can see the webbing I'm talking about. This is just, classic, typical spider mite symptoms. In almonds, this is kind of what you get. You can see the webbing on the top left, but more common on the right. You can see how everything's just kind of turning brown. You can see some webbings forming at the tips of those particular shoots. At the same time, that particular tree, from a photosynthetic standpoint, is still intercepting 100%, pretty close to that of the sunlight that's coming to that tree. So, when I see a tree like that, my response is, that's not pretty, but that's not a big deal. The question is that as bad as it gets, or does what you see in that picture turn into complete defoliation, which obviously is a major problem? So there's balances there between what you can and can't tolerate. Now, going into cultural practices a little bit, there are some conditions that promote mites. And the first one of those is stress. So anytime you have a stress to the tree, and this isn't just in almonds, I mean, this is true in cotton and pretty much any other crop that has spider mite problems. Anytime you have a tree that's short on water or that nutritionally is not right, there's just something about the chemistry or physiology inside of that tree that the mites just know something's not right. They think maybe the tree is going to senesce, they're running out of their chance to eat, and they reproduce quicker, and your problems flare. And you can see my grape case study here. That's just to remind me, situation down in Kern County where we'd have cherries next to wine grapes, next to table grapes. All three of those are great hosts for spider mites. And right next to that, almonds. And it was really interesting because you go out to the wine grapes, and the mites are a problem when they're doing their regulated deficit irrigation. You got to the almonds, and the mites were a problem after hull split when the trees were water stressed. You go to the other side of the intersection in the table grapes, where there's never any stress and there was never any mite problem. And then you go over to the cherries that have their full crop load going on in May and getting picked in May, maybe getting neglected on water a little bit. In that case, the mites problems were at the end of May and June. So, the same mite species, 4 different crops, at the same intersection, and the mite blowups, if you want to call it that, occurred at 4 different times in those 4 crops. Okay, that's not related to biocontrol. That's not related to temperatures or anything like that. That's literally when is the plant stressed. So that helps explain why post-hull split is really the time in almonds that we get mite problems. Dust will also promote mites. Dust just gives them lots of little attachment points to make their webbing. Instead of just sitting on the surface of the leaf, they can make sort of high-rise, multi-level homes and put all their eggs and live within all that webbing. And then the other thing that can really make mites a problem is lack of natural enemies. So mites are one of those cases where there's lots of natural enemies. They're very good. But if you kill them or do something that caused them not to be there, your mites will blow up. There's no such thing as an almond orchard without mites. And the point with that being, the difference between a good and a bad mite year is not whether or not mites are there. Okay, the difference between a good and a bad mite year is the level of biocontrol present. So we'll spend some time talking about that in a minute here. So just to sort of summarize the rest of the presentation here, IPM for spider mites, there's sort of, these are what I'll call the 4 major points that you'd have in a program. So first of all, with spider mites, you want to monitor weekly, and you start that in March. Around now, if you haven't done it already, you should be starting. And that's a presence absence sampling for mites. You also want to be monitoring for natural enemies. So we'll talk about what those are. You need to be using thresholds. We're going to spend a good bit of time talking about how thresholds were developed and how to use them. And then if you do need to treat according to a threshold, you want to do that with a pesticide that doesn't kill the beneficials. And that also applies for navel orangeworm and other pests where you're spraying for those, that you want to make sure that your navel orangeworm or your stink bug or leaf-footed bug sprays, whenever possible, can preserve the natural enemies that you want for the spider mites. So just to start off on the sampling, this is pretty much straight from the University of California's pest management guidelines. What they say is prior to July 1st, you want to focus on hot spots. That's edges of the tree, or edges of the orchard, crotches of the tree, because that's kind of where the mites are to start with. You want to pick random leaves. I know this is hard. Our eyes are kind of trained that when we walk up to the tree, we're looking for damage. Okay. Well, if you go up to a tree and you're looking for the one leaf on the whole tree that has mites, the thresholds won't work. Okay, that's not what they're based on. They're based on random leaf selection. So random leaves, 15 per tree or within a few tree area, at least 5 trees, and presence absence for mites, presence absence for predators. It actually goes pretty quickly. And there's a form that's available on the UCIPM website. If you don't have your own system, this form is pretty good. I don't recommend using it every week just because I realize it takes time to fill out a form. If you've got 0 mites, don't waste your time. If your tree's defoliated, you missed your spray. You missed your timing. All right, you don't need the form. But during those critical weeks where you're trying to decide, do I spray or do I not spray? I actually think this form is very, very useful because it'll give you, it'll show you the numbers that you have of beneficials and of mites, and it's got the thresholds on here. So I'm not going to go into too many more details about this, but I just want you to know the form is available and can be very helpful, both to see whether you should treat in a current week, but also if you fill out this form each week for 2 or 3 weeks, right when you're making those mite spray decisions, it really allows you to see over time how much or if the mites are going up, but also how much and if the beneficials are going up to match it. Both those pieces of information are important. Now, as I said, when you're monitoring, you also want to look for biological control. So who are these critters that you want to be finding? So first of all, you want to be looking for predatory mites. Any literature you find on spider mite management in almonds, you're going to find it's just all about predatory mites. Historically, these have been the most important ones out there. And if you look at the picture on the top left, okay, that's a predatory mite. That's the Gallandromus occidentalis. And you can see that it's sort of teardrop shaped. It's pointed on the right and round on the backside. Okay, that's common for predatory mites. You can see that it's sort of an amber color. That's also, in fairly uniform amber color. That's common. They're very shiny. They don't have a lot of hairs on them compared to spider mites. So you can see that sort of reflection of the flash or sunlight on that picture on the top left. And predatory mites can be very effective. So you want to be watching for them. On the right-hand side, on the top, you can see the predatory mite on the bottom, feeding on the spider mite above it. And in that case, the spider mite's a good bit bigger, but they can do that. The predatory mites are also very good at eating the eggs of the spider mites. Obviously, they can't run away. So nice little juice sack, if you want to call it that. So predatory mites, number one. Oh, the other thing about predatory mites is they're very active. So you'll commonly see them running around on the leaves, whereas the spider mites will typically just be sitting there. Second predator you want to be looking for. This is one that can be around in the spring, but it's very, very common late in the season, sometimes almost too late. Nevertheless, you want to look for it. And this is called the spider mite destroyer beetle. So it's actually a ladybug. It's about maybe a third the size of what you would normally think of as a ladybug that you just, the orange ones or reddish ones you'd find all around. So on the top there is a picture of the larval stage. It crawls around the leaf and eats mites, eats a lot of mites, really loves to eat the eggs. The middle picture on the left is the pupa, and those will be attached to the leaf. And then on the bottom left is the adult beetle. And you can see shiny black. And just to give a size perspective on the top right, you can see the size of that beetle compared to the size of an almond. So relatively small, but they're not hard to see at all. Now these females, they will live 1 to 3 months, so very long lived, and they'll lay about 100 to 200 eggs during that time period. So yeah, and then about 3 weeks they go from egg to adult. So they don't have the quick reproductive rate that a predatory mite does, but they've got some longevity. And once they show up, they're very effective. Now for some generalist predators, green lacewings, I hope you're all familiar with these. You can see the picture on the top right of a stalk that has the egg on top. Even after that egg hatches, those stalks stick around. So when you see those, that means you got some lacewings. The center right is the larval stage. It looks sort of like alligators, except their mandibles go side to side instead of top to bottom like an alligator. They'll use those mandibles to grab a mite and they'll suck it dry. And go to another one, suck it dry. Go to another one, suck it dry. They're actually very, very nimble and agile with those mandibles. It's pretty neat to watch if you have the chance. On the bottom left is the pupa. These pupae are usually underneath the bark. So you're peeling away chunks of bark and you see that sort of white, cottony, round ball. Again, that means you've got lacewings. And the bottom right's the adult. The adult stage does not feed, but obviously they reproduce and they make more babies, so we like them. I want to keep them around. Another predator you'll find is called minute pirate bug. This is most common if you're near alfalfa or other field crops. The pirate bugs really love those crops or next to corn. The nymph stage is on the left. It's kind of translucent. It has a little bit of an orange pattern in the middle of the body. And then on the right side, you can see the adult minute pirate bug. It's black and white sort of checkered pattern. And then lastly is sort of a relatively newcomer in importance within the almonds, which is called six-spotted thrips. And I'm going to spend a good bit of time talking about six-spotted thrips in the rest of this presentation because it's become what really matters. So the six-spotted thrips is a predator of spider mites, period. Okay, that's all it eats. And the six-spotted thrips, it thrives in hot, dry climates. Can eat about 50 eggs per day. Okay, that's lab data at 86 Fahrenheit. The population can quadruple in a week. So literally they can double twice in 1 week's period. Now they're what we call thigmotaxic, which means they love to get into webbing. There's a lot of predators that just don't want to get into webbing, but they absolutely love it. And they're cannibalistic. So that means that if you have a whole bunch of thrips and they eat all the mites and all the mites are gone, they'll start to eat each other. On one hand, that's not good if they eat each other because they're predators. At the same time, eating each other allows them to survive until the mites show up again, in which point, they'd be ready for that food. Now, with this particular thrips, about 90% of the population is female throughout the year, which is good because the females are larger than the males, they eat more, and they're the ones that reproduce. And they've pretty much replaced the predatory mites as the most important predators out there. And what we think is happening is that the six-spotted thrips eat the spider mites, but they also eat predatory mites. They're not, I guess to a thrips, they're both a chunk of meat. The eggs are both little juice sacs. I don't think they care. So they will eat both. So the negative side, the predatory mites are going down, but predatory mites, if they serve as a food source for the thrips and the thrips are eating the spider mites, it's all good. So, okay. Miranda, real quick, can you just give me a check? I just want to make sure the audio is coming through and everything.
Yep, you sound great.
Perfect, thank you. I can't see anybody else or anything, so I just want to make sure I wasn't talking to myself for 10 minutes. All right.
Yep, we can hear you.
Great. Okay, so I want to. So why are thrips different? Why are there so many now? And I just want to point some things out for those of you that have been around, for a couple of decades. Let's go back 15 to 20 years. Well, guthion, it's gone. Okay, supercide, it's gone. Lorsban is now gone. Dormant treatments, they're mostly gone. And the dormant treatments that are left, a lot of it's just oil. Insecticide during bloom, mostly gone. A little bit of BT out there, but that's the only insecticide that should be going on in bloom at all. Pyrethroid use is declining. Navel orangeworm's developing some resistance to it. Pyrethroids aren't being used as much as they were. And then there's other things as far as production practices, like flood to precision irrigation. It used to be that the trees were super hydrated and then dehydrated and then super hydrated and dehydrated. They'd go through these hydration swings that would cause spider mites. That tree stress causes it. Now that there's these precision irrigations, each variety's on their micros, you're watering trees before your nuts are even off the ground once they're in windrows. All those abilities help change the dynamics for mites. So, compared to 20 years ago, San Jose scale used to be a massive issue. And now it's pretty negligible. Peach twig borer used to be a major problem. Nowadays, there's plenty of peach twig borer out there. If you put a pheromone trap out, you'll catch lots of them. But when you actually go to the huller and look at percentage damage at the nuts, it's becoming more and more of a non-issue. Ants, ants are under great control. We're not spraying Lorsban on the ground to control ants like we did 15, 20 years ago. The baits are great. For navel orangeworm, we're using mainly Intrepid and Alticor with a few other products. Also main disruptions in there. So basically the almond industry's gone green, very much so. And all of these shifts are affecting the number of natural enemies for spider mites. So it's changing things. So with the thrips around, one of the issues with the thrips is there's no way to monitor for it. I mean, you can kind of look for them, but it takes a lot of time to see them. And from a scientific standpoint, if you just walk around an orchard and say, oh, look, I saw one. Oh, look, I saw one. Okay, well, that's not very scientific from a thresholds or monitoring standpoint. So one of the first things we did once we realized how important these thrips were is try to figure out how we could monitor for them. And in lots of different crops, sticky cards are used to monitor thrips, greenhouses, onions, I mean, all kinds of stuff. So we took a whole bunch of traps. You can see the picture on the right. In this case, this study had 7 different cards, but I think we did 9 different cards. Multiple locations. We looked at different sizes of them, different shapes, different colors and so on. And the bottom line was trap type does matter. Okay, the 7 traps you see there all got very different numbers of thrips. Even among the 4 yellow ones you see there, some of those yellow ones were great and some of those yellow ones were worthless. Okay, so I really want to highlight that. If you're going to monitor for thrips, don't just go out there and just pick a random card off of whatever internet site, because that's not going to work. Okay. What we did is we went through all these. We looked at what they cost, how easy they were to use, how effective they were, and we made a choice. And there was actually a very clear winner when we did that work. It's a trap that used to be called a yellow strip trap. Nowadays, it's called a predator trap. It's a product, Trace makes it. We purchase them through Great Lakes IPM. They're like a quarter a piece, pretty cheap. But you can see the middle picture there. It's a little bigger than a 3x5 card. And what we do is we stick a binder clip to it, and then we use a paper clip, just a large paper clip that we unwind, and we hook the paper clip to the binder clip, and then hook the paper clip to the tree. Just kind of hook it on. And so you go out, you hang this, leave it there for a week, and you can see the 3 pictures on the right. When you come back, the thrips are on there. And you can kind of see those little brown guys on the top picture there on the right. You can see it blowing up a little bit in the middle picture. And then when you zoom in on the bottom picture, they tend to land feet down, which means wings up. And those 6 spots that you see there are on the wings. So with a little bit of training and a naked eye, I have no problem. I've got good eyes. I have no problem looking at a card and just counting the thrips. At the same time, if your eyes aren't great, if you get a hand lens out, you'll have no problem seeing those 6 spots on those wings. So yeah, you just leave it there for 1 week and you come back and you count it. And it's that easy. Much easier than any other method that we looked at. So once we developed a trap, now we had some opportunity to start doing some things with it. And back in 2016, this was a kind of fun where we had 3 sites in Kern County, and this here shows the number of mites per leaf. So over time. So, in July, you can see the mites started picking up, the mites got up and up and up. And then the mites did this in all 3 sites. And, usually when you look at these kind of data, it means that the grower went in and sprayed a miticide. And therefore you got the drop very quickly over a week's period. The interesting thing is none of these fields had miticides. All 3 of these, this was just naturally occurring. And it occurred in August. And, certainly there were still plenty of leaves and conditions were great for mites in August. So what happened? Well, these are the data every week from 6 spotted thrips in the traps. And if you start lining these 3 fields up, what you can see is the mites start going up. There's a little bit of a lag time. And then you see the blue line start going up. And as the blue line goes up, you reach a point that all of a sudden the blue's going up and the green line crashes. And essentially the mites go away. And so we started realizing, well, hey, these thrips are pretty cool. Look at this. This is what you study in graduate school, right? Where you've got the prey, and then the predator has a lag time, and then the predator eats the prey, and you get this predator-prey relationship going on. One other thing to point out is those yellow traps that I showed you also collect adult beetles. So the spider mite destroyer beetle, and the red line shows them. So you can see there also, in that case, the lag time is longer, and they do increase in number. By the time their numbers increase, the mites have kind of gone away. So they come a little bit late to the party, at the same time, I don't want to ignore them. They are important in biocontrol and certainly played a role in the green bars you see at the top. But so we saw this in 2016 and, in other years. And what we did is we started taking all these fields and putting them all together to see a relationship. Now, we're dealing with different years, different times of the years, different dates. So the first thing we did is we said, let's just go to all these fields and pick a day that there's 1.4 mites per leaf. Okay. We picked that number. It's based on literature. I don't need to go into all the history, but we basically said, let's just call the day that we hit 1.4 mites per leaf day 0. And then let's look at C, take all these fields, put all the data together and see what were the mite populations and the thrips populations up to 3 weeks before that, and then up to 80 days after that point. And you can see what we get here. And this is absolutely gorgeous chart. I hope you appreciate the work that went into this, because this to me is very telling of what's going on. You can see the solid bar. That's the spider mites. You can see them start going up. You can see there's that lag time. It's about 2 weeks. And then you see the thrips start going up. And as the thrips go up, the mites come down. When the mites disappear, right about day 40, 45, once the mites are completely gone, the thrips run out of food, and you can see the thrips population crash when they run out of mites to eat and then run out of each other to eat, to the point 70 days out that there's neither. So this is just classic biocontrol predator-prey relationships. Once we showed that this is happening, now we can start doing some really fun math, okay? Because we've documented that this relationship is real. Now, the other thing we're able to do with the 6-spotted thrips, before I get into the more, following up on that previous theme, is we did, for multiple years, traps all year long. And this is just 2 years worth of data. But you can see is every April and May, so it starts about mid-April and goes about mid-May in Kern County. If you're up in the northern San Joaquin Valley, it's delayed just a little bit. Just imagine that April-May hump shifted over maybe 10 days to the right to where it's sort of end of April through the end of May. But every single orchard we go to in this April-May period, we get this pulse or blip or whatever you want to call it, period of adult activity of the thrips. So what we're pretty sure is going on there is that's the thrips that are overwintering. So they overwinter as adults, and then in April and May, those adults come out and look for something to eat, which is perfect because we know that spider mites around the end of March and beginning of April is when they start coming out. So right when the mites are showing up in April and May is right when the overwintering adult thrips come out and eat them up. And you can see here by the end of May, the thrips disappear. That's because the mites disappeared. They ate them all. So we see that routinely. And then what we're seeing commonly now is that June and July, there's no mites anymore because the thrips ate them up. And the thrips, of course, without any food are really, really low. And then once we get to hull split or beyond, out in July, the mites just naturally start coming back up. It's hot, it's dusty, you're pulling back some water, doing a little regulated deficit irrigation for hull rot and to try and synchronize your split and so on. All those conditions promote the mites. As those mites go up, you can see here the thrips respond on this chart. So, the solid bar is 2017, the dotted line is 2018. You can see that those aren't superimposed right on each other. That's because in our particular trials, the mites showed up earlier in 2017 and showed up a little later in 2018. So the thrips are matching the mites. So 2 periods of activity, okay? One in April, May. We're going to come back to that because that's the sort of prophylactic May spray topic. And then, the thrips in this August, September period, that's what you're going to have during harvest. So we'll come back to that. Oh, one other thing with these charts is we're able to look at population doubling time. So I'm just showing you 5 sites here. We've got about a dozen sites total, but at the end of the day, the spider mites are taking about a week to double their populations. The six-spotted thrips, here it says 3.4 on the bottom right. When you put all the sites together, it's about 4 days is about what it takes to double their population. So very, very quick increases can be seen. So we know what, we've got these thresholds for mites, what we'll kind of go into, and then we've got, the thrips to take into account. But just conceptually, let me show you what we're trying to do here. So this is, I think it's 9 different fields with no miticide treatments, and it shows you what mite populations look like. Now, in all case, the 0 right in the middle, that was the first day that we got what we would call a threshold number of mites. But you can see, before that, the mites just kind of go along. They're there. There's a few, there's a few, there's a few. And then there's just something about that treatment threshold timing that once the mites get to that population, they just take off. And that's what scares people to death. This explosion of mites that can just lead to defoliation within a few weeks. So, what's the goal of a treatment threshold? Well, the goal of a treatment threshold is that you treat right before that big increase happens. Now, we know that from the time it takes you, from the time you decide to treat until an actual treatment is made, it's common for that to be a week. So let's just assume that's 7 days. So that means we have to define an action threshold. So what is the mite population 7 days before a treatment threshold is reached? Let's call that an action threshold. That's the day that you have to make a decision to spray so that you can spray at the treatment threshold to prevent those numbers from going up. Now, what happens if you wait too long to treat? Obviously, you get in this nightmare situation on the right and you risk defoliating your trees. Okay, we don't want to do that. But I want to also point out there is a risk of treating too early. Okay, where the mite populations 4, 3, 2, and 1 week before the treatment threshold is reached, those mites are predator food. If you want predatory mites and you want lacewings and you want six-spotted thrips and you want all these predators to be helping you out, including all the way through harvest, you can't remove all their food. Okay, you've got to give them something to build up on and something to feed on. And so that's why we really want to wait to treat until you get to a threshold. Because if you treat before that, you starve all your predators. And then when the mites come back, there's nothing to control them. You literally can make your mite problems late in the season worse by treating too early for mites earlier in the season. Okay, so treatment thresholds have been around for a while. The first work in this area was done by Frank Zalem. So Zalem and Wilson back in 1984. I have a fun time whenever I mention this paper to Frank because I'm assuming he's still active and still involved up there at UC Davis, but I do have to tease him about how long ago that paper was. Just take a little cracks a little bit about his age, but it's neat to me to look back 1984 that the person that did this work is still around and we can still chat about it. It's pretty neat. But in that work by Zalem and their team, what they did is they went out to a bunch of trees, got a whole bunch of experts that have been around for a long time, and they looked at a tree and said, does that tree need to be treated or not? And they basically just made that decision on a whole bunch of trees. And after they decided whether or not the trees needed to be treated, people went through, probably Frank, back in grad school, early career, went through and counted all the mites and then determined whether or not there's predators. And they crunched all those numbers and they wrote a couple of papers that are very good. And what they determined was if there's no predatory mites, okay, that was the main biocontrol back then. If there's no predatory mites, you need to treat if 22% of the leaves are infested. Okay, and if there's predatory mites, if biocontrol is present, you should treat if there's 43.6% of the leaves infested. And that's based on some calculations and curves. So again, 22% if there's no predators, 43% if predators are present. And they developed, based on that, a sequential sampling plan, which is the monitoring form you see on the right. It is the way you would access that. And that's well documented in the literature. So since 1984, this has been what the University of California has recommended. I've used it. It's been very successful. At the same time, that's 1984. So, 94, 04, 14. Okay. It's now 35 years later. And a lot has changed in 35 years. So we asked the question as, are these thresholds still valid? And, we wanted to take a look at that. So we took a very, very different approach. Completely different approach than what Xalem did. And in this case, we took 12 different orchards, large orchards, and didn't make any treatments in them. We tracked the mites over time to look and see how they built. And then we synchronized all of these fields by saying, what was the first day that we got to 1.4 mites per leaf? Okay. You can see on the bottom right here, here's the mite populations in those 12 orchards, 3 weeks prior to and 3 weeks after reaching a treatment threshold. And you can see, as was shown before, prior to the threshold, the mites just kind of slowly come up. After that threshold, the 0 day, the mites go crazy. And that's what we're afraid of. So we made this curve, put all these data together, and we sort of defined, so this line right here, where this line is at a 45-degree angle on the curve, we just called that the treatment threshold. Okay, we said prior to that number, we're not overly worried about mites. Prior to that number, we should be freaked out about mites. Okay, so that's the day that a spray should be made to make sure that the mites aren't a problem. And then we said, okay, let's come back 7 days because we know how long it takes to make a treatment. And we define that as the action threshold. So the day that a decision should be made to treat. And the results of that, what it showed was the action threshold. So you should decide to treat if there's 1.4 mites per leaf. That way you can prevent the treatment threshold, or your treatment is made in time for the treatment threshold when there's about 5 mites per leaf, so that all that process is complete before that line gets any higher and before any economic losses occur, which is somewhere up towards the top of this chart. We don't know where that exact is, but 20 mites per leaf for a week or so, 30, those aren't a big deal if that's as bad as mites get. So we're essentially preventing all of that using this action threshold and treatment threshold. So, well, 1.4 mites per leaf, what does that mean? Okay, we know you don't count leaves or count mites. So when we ran all the calculations, 1.4 mites per leaf is the equivalent of 38% of leaves infested. Okay, you remember the Xalem threshold that I showed you a few slides ago? Frank Xalem and et al. Said that if you've got biological control present, you should treat when there's 43.6% of leaves infested. So I don't know about you, but for me as a researcher, to think that somebody 35 years ago used one method for determining thresholds and came up with 43%. And 35 years later, a different group of scientists using a completely different methodology came up with the number 38. I'm sorry, but research just never comes out that pretty. For me to see that what we determined and what Frank determined 35 years ago are both basically 40% leaves infested, to me is just phenomenal and exciting. And I didn't want to tell Frank that he was wrong. I'm really glad that my number came out right on top of his. But, from your standpoint, as far as credibility, this 40% of leaves infested, it's not just made up. I went through pretty good details here how it's developed. I hope you realize that 40% number, it's solid. It's based on 2 researchers 35 years apart, lots of different fields, 2 methodologies. I personally have a lot of confidence in that particular number, and I hope that you do too, because I'm very familiar with how much work went into it. said, unpause here, 40% of leaves infested. That's where a treatment should be made. But let's also remember this 40% of leaves infested is only taking into account the mite population. Okay, just the mites. Well, this is integrated pest management. This is the 2020s. We can do better than that. And, integrated pest management, we need to think about the biocontrol. So what we figured out is we kind of use this adage now, if you want to know how many mites there are, count the mites. But if you want to know how many mites there are going to be, count the thrips. Because that's what we're finding out. If you look at this chart on the bottom right here, the x-axis, this is the thrips to mite ratio. Okay, so where you see that arrow at the 400, that means that there are 400 six-spotted thrips on one of those sticky traps during a week for every 1 mite per average on a leaf. Okay, that's what 400 means. The y-axis, this is the change in mite density over 7 days. So a value of 30 towards the top means that there's 30 times more mites in 7 days than there are today. Okay, and obviously as you approach 0, that means, less, right? So what you can see here is what you would expect. If you have an infinite number of thrips to mites, the mite population just obliterates it. It's gone. And as you get on the very left-hand side of this bar, of this chart looking up, as you have no thrips per mites, you can see that the mites explode. You get these outbreaks. So, sometimes people will say, hey, David, I've got 3 mites per leaf. Should I treat? Well, that's an unfair question. If you've got 3 mites per leaf and there's no predators, yes. But if you've got 3 mites per leaf, you may have had 20 the week before and you're at the tail end of a crash. Okay, so again, it's not so much the number of mites per leaf that drives the change, it's how many predators are there and what are those ratios. So we started doing a whole bunch of work with that. And so you can see this chart here. If this is the exact same chart, I just took the x-axis and the y-axis and put them onto log scales. So now we can try to make a line out of them. And what this line shows is that mite populations remained unchanged in a 7-day period if there were 0.42 thrips per card per week for every 1 mite per leaf. Okay, I'm going to say that again because I know it takes a second to kind of think through, but if you've got 0.4 thrips on a card in a week for every 1 mite per average on a leaf, you're going to break even. Okay, if there's more thrips per mite than that, population of mites is going to go down. If there's less thrips per mite than that, the population of mites is going to go up. Okay, so we developed this kind of threshold or this number here. Now I realize there's a problem here because you're not counting mites per leaf. Obviously, you can't count mites in the future. We're doing all this research in hindsight. So I'm just going to interpret these data for you. And what do these data mean? So first of all, let's talk about May sprays. We already know that in May, you should not treat unless you have 40% of the leaves infested. Okay, the Zalem threshold, the Haviland threshold, both of those agree 40% of leaves. If you're under that, don't treat. Okay, anyone that's doing preventative or prophylactic sprays for mites, I'm with you if you say that was necessary 10 years ago. I'm not with you if you say that's necessary today, because it just simply is not. So at this 40% of leaves infested, if you take that number that we had from the last slide, okay, it showed that mite density will not change if there's 0.6 thrips per card. Okay, that's per mite per leaf. I realize it's getting a little complicated here, but on the third bullet here, what this means is if you're finding an average of 1 thrips per card in April or May, and you crunch all those numbers and line up all those thresholds, if you're finding 1 thrips per card in April or May, you do not need to treat. Okay, so the fact that you're under 40% of leaves infested, combined with finding 1 thrips per card on average, both of those doubly tell you to do not treat. And for the record, we've now monitored more than 20 orchards in April and May. Every single one of those 20, 100% of those 20 orchards had more thrips than this threshold. Okay, every single one. And that's sites down in Kern County, that's sites up in the northern San Joaquin Valley, sites up in the Sacramento Valley. You can see on this chart on the right, our averages in April and May, we're commonly in the 20 to 30 thrips per card per week. And it only takes 1 to control the mites. So unfortunately, I think what's happening is mites are coming out in the spring. The thrips are coming out and eating them all right when people are putting on their miticide sprays that are completely unnecessary because the thrips would eat them all on their own. So just, yeah, the days of preventative prophylactic mite sprays are done. We shouldn't be doing those anymore. There should be no treatments in April or May unless you're at 40% of leaves infested and you're not finding any thrips. Now, a couple of exceptions I do want to highlight. First of all, non-bearing trees are totally different. Okay, so don't use these numbers on the non-bearing trees. You just kind of have to treat them separately. The other thing is, if you plan on killing the thrips, okay, if you're going to go in with a broad spectrum pyrethroid or something for leaf-footed bug in the beginning of April, that obviously is going to change things. You're not going to find the thrips that you should be finding. In that case, use the 40% of leaves infested threshold. It's still valid. But again, in that case, it's not a prophylactic spray. It's a spray based on numbers. So what about hull split timing? Hull split's a little more problematic because what we're finding is that the mites reach a threshold at a time you can't treat. Is kind of what normally happens. So what you're trying to do is instead of saying, I'm going to treat when I hit a threshold, you have to look at when your hull split spray is, or if you're doing 2 hull split sprays for navel orangeworm, you pretty much have to look at the mite populations at those times and say, number 1, am I at a threshold? But number 2, are the numbers looking like I'm going to hit a threshold in the next 2 or 3 weeks when I'm harvesting and can't spray? So to respond to that scenario, what we did is we took all the numbers and we completely redid the whole model. Instead of saying how many mites requires a treatment, we said, based on the number of thrips per card, what is the probability that we will reach a threshold anytime in the rest of the season? Okay, and what we determined on this bottom left slide here, or bottom left bullet, probabilities showed that if you had 3 thrips per card in a week, you're not going to have any change in mite density within 14 days. 3 cards per week, 77% of the time, within 14 days, the mites would be lower. Okay, so that three number is kind of like if you're getting 3 and trying to decide if you should treat, you're in pretty good shape. You pretty much don't need to treat. But based on you may or may not. That's kind of where you draw the line there. If you're up in the 5 range or 6 thrips, walk away. I don't care how bad your mites are. The thrips are just going to take them over. So again, 3 thrips per card per week, break even point, 5 to 6 per week. You will never hit a threshold in the whole rest of the season, all the way through harvest. Okay, I'm going to wrap things up just talking a little bit about insecticides. We're at 9 o'clock here. Insecticides, or miticides in this case, are valuable. This is a picture from 15 years ago now in a trial I did in a plum orchard, but it just shows 1 tree was treated, 1 tree was not. That's what a miticide can do for you if you need them. Now, there are a lot of miticides out there. So just to kind of walk through them really quickly here, I put them in order of their mode of action. But you've got Agromec. Agromec's best before the leaves harden off because it has a translaminar activity that moves into the leaf. And the mites ingest it when they feed on the leaf, and then they die. So Agromec's one of the most common products used in the April or May windows before the leaves harden off. It's also one of the, if not the least expensive miticides, so gets a lot of use. The one drawback, though, to Agromec and the other abamectin products is that they are toxic to six-spotted thrips, so I want to be a little bit careful there. One perk, though, is that Agromec also has activity against leaf-footed bug. No residual, but just some direct contact activity. So, so if you're in that early season leaf-footed bug and you need to just knock back some leaf-footed bug adults, but also want a miticide, and you're going to kill some thrips in the process, but it makes sense to use it. All the rest of the miticides have no impacts on biological control that I'm going to mention here. So you've got growth regulators, Apollo and Onager. In this case, they're causing the eggs not to hatch and has some nymph activity. Zeal is also a growth regulator, but it works very differently. It inhibits the molting of juveniles, and it has some contact activity on the eggs. Mite is pretty much an obsolete product, but it's still registered, so I listed it here. Acromite and Vigilant, these are both, anything you see here that says meti, this is something that affects the mitochondria. It's a direct contact kind of product. These are your knockdown products. So Acromite and Vigilant, contact materials, work very quickly. Fujimite and Magister, likewise, are meti products, work on contact. If I skip Envidor at the bottom, Nialta is also a meti product, good knockdown. And then Envidor is a growth regulator also that affects the ability for them to produce fat bodies. So all these are available, and I'm just going to say they're all good. These products are all effective, and we've done lots and lots of miticide trials, and they all sort of look like this, where you can see the black bars: Acromite, Envidor, Fujimite, Magister, Nialta, Onager, Vigilant, Zeal are all down in the look good range. All those with oil, by the way. In this case, I put Grandiva with oil. You can see it was comparable to oil by itself there. There's a lot of oil products out there. I'm not really showing those here, but oil is quite effective on mites. You don't always need a miticide. And then you can see the red bar, that's the untreated check. So this is very typical that you got an untreated check, oil by itself, 50% reduction in damage or reduction in the populations is common. And then oil plus the miticides are always somewhere down below that, but most of the products tend to have similar efficacy. Here's another study. You can see Zeal, Envidor, Onager, Apollo, Fujimite, Acromite all did very well. You can see what oil did there. In this case, Vendex didn't really work. Sometimes that happens, but you can see how good these miticides all are compared to the untreated check. Here's a trial in 2018. Same kind of thing. Banter, that's a generic Vigilant. So, Banter, Vigilant, Envidor, Fujimite, Canamite, Magister, Nialta, Onager, all look great. You'll see Biomite, Cinerate, and Tetracurb. Those are three biological, organic kind of products, kind of oil-based. But you can see they did fairly well, a little bit better than oil by itself. And that's kind of what we see with the biological products. They tend to be a little bit better than regular oil, but typically not quite as good as the full-blown traditional miticide. Nevertheless, acceptable control. You don't always need the best miticide. What you need is a miticide that does a good job at knocking back the mites, that preserves the beneficials, and the beneficials clean up anything that's left over. That's just kind of how IPM works. So I'm not going to split hairs on miticides. I'll just say they all work. They're all good. Certainly if you're trying to put out a fire, don't go with a growth regulator. Go with one of the meti inhibitors. Likewise, if you're treating at a normal threshold, The numbers we've talked about, any of those products are just as good. Some work a little quicker than others, but all recommended. One thing though is coverage is important with mites. And I mean, I cannot overemphasize the importance of coverage. Most miticide failures are simply situations where you've got a miticide that works on contact that doesn't come in contact with the mites. And the most common problem is driving too fast. So you really need to have the sprayer at 2 miles per hour. And that's, just for 30 years, research trials have continually over and over shown that at that 2 miles per hour with a fan, that's the speed you need, that the fan and the product is going to move into the tree, that you're going to displace the air in the tree, and that you're going to get coverage in and throughout that tree. Okay, if you're going faster than 2 miles per hour, the product tends to sort of fall into a sort of vortex behind the equipment, and you don't get full displacement of air in the tree. You're going to get poor coverage, and you're going to have mite problems, and you're going to blame it on a miticide that's not to blame. Okay, as far as water volumes, early season, when there's not a lot of foliage, the leaves are tender, there's not a whole lot of dust, 100 gallons per acre, Agrimec applications, that's fairly common, that can be effective. But if it's, if the leaves are dusty, or if it gets later in the year with larger trees, bigger canopies, you need to have 200 gallons per acre to get decent coverage with any of the miticides. Now, fan speed, I'm just going to put here, enough fan to displace the air in the tree, but not to blow products through to the next row. And I mention that because this is really an issue sometimes in baby trees, where you've got the air blast sprayer all set up for your 15-foot tall trees, and then somebody uses the same equipment. Maybe they turn off the top 2 nozzles, but they use the same fan speed and everything else on these baby trees, and they just blast the fan sideways. You can see product go in 2 or 3 rows. This isn't a spray coverage talk, but I'll just tell you, when you're blasting air past or over the surface of a leaf, it's kind of like the wind going over the surface of a wing on a plane. The pesticide just goes above and below the leaf, never really gets on the leaf. So more air is not always better. Okay, you just want to displace the air in the tree and have the product settle there. And then, nozzle types, again, this isn't a spray coverage talk per se, but you want some kind of nozzle that's going to give you the best coverage possible, including the tops of the trees. So, maybe that means, some air induction nozzles, particularly on the tops. Not an expert in this area, but just point out it matters, especially if you're a PCA. I've talked to a lot of frustrated PCAs that say, I want you to drive at 2 miles per hour, 200 gallons of water, this kind of coverage and so on. And then the applicator goes out there and just does something totally different. And then the PCA's got to come back and figure out what went wrong. So please follow up on the equipment, make sure it's good. So with that, just to wrap things up, summarizing what I think a grower should be doing, or PCA, every week monitor, no doubt, that's a given. Okay, you want to be doing everything possible to conserve biocontrol. So thrips are your best friends. I think everybody's probably seen the situation where the mites blow up during harvest. You can't do anything about it. You get frustrated. And then all of a sudden you come back 7 days or 14 days later and the mites are just gone. Okay, that is 6-spotted thrips. Okay, that didn't just happen naturally. So the more you can do to keep these beneficials around is the better off you're going to be. We highly recommend using thresholds. I put 25% to 40%. Really, it's 40 these days. Okay, that 25% of leaves infested, that threshold is still valid if you have 0 biological control, which was common in 1984 when those thresholds were developed. Nowadays, I don't know any almond orchards that have 0 biocontrol. It's kind of impossible to have 0 biocontrol with our normal programs these days. So for that reason, stick with the 40% of leaves infested when you're making your decisions to treat. Okay, avoid prophylactic sprays in May. There's just no place for those anymore. They're not needed. unless you plan on killing the beneficials and are responding to that. Yeah, it's a little different situation. And then, at hull split, in most cases, the grower is going to spend the whole year monitoring for mites, not really find any. And then they're going to get out to hull split or a post-hull split spray timing. They're going to see mites start showing up, and they're going to have to make a decision. And do I spray or do I not? And that decision is going to be based on a projection of whether or not you think a threshold is going to be reached during harvest. And that requires monitoring of mites, a little bit of prediction, but really it has a lot to do with thrips. So, if there's no mites, don't spray. But if the mites are building up, consider a spray. But if the mites are building up and you see there's 3 thrips per card in a week, you're going to be good. You can walk away. In most cases, if you're in the 5 to 6 thrips per card range, guaranteed, there's nothing you need to do. And I've had several people call me that just say, David, I put on my, I put a miticide in my hull split spray. It's 3 weeks later. I'm starting to see mites build up. I'm concerned because I'm going to be shaking my nonpareils and, whatever. Should I do something? And I'll always tell them, put a card out, because that's what you really want to be looking at. And they'll hang a card and they'll call me and they'll say, hey, the card's only been out for 4 days. I need to make a decision, but I've got, I got 13 6-spotted thrips on the card. And I'll just tell them, hey, walk away, go sleep at night. You're going to be good. And sure enough, I'll email them, 7 or 10 days later. And they're like, hey, wow, the mites just kind of disappeared. And now I'm getting, 4 times as many thrips on the card. And I just say, congratulations, you saved yourself a $60 spray, and you just got to see biological control in action. Congratulations, that's integrated pest management. You are a participant. It's fun when those situations happen. So that's the guidelines we can give as far as treatment decisions. There's an art to it as well the science. Obviously, if it's going to be 110 degrees next week versus 80 degrees, that changes things. Obviously there's, free rides to consider. But hopefully with the principles that I've talked about here, I think people have all the information they need to make some very educated decisions about mite management. This is a case where IPM can rule and should rule. Mites are also one of those areas. The almond board's working really hard to promote the almond industry as, sustainable, using integrated pest management, green, healthy, and so on. And mites is just an awesome case where the entire industry can show that it's using integrated pest management principles, relying to the most extent possible on biological control, that when miticides are needed, that it's being done prudently based on thresholds, and keeping the natural enemies around. That's something anybody can sell. And I hope everyone can be involved as much as possible in that, and let me know if you have questions in the process of doing so. So thanks for being on this morning. I hope this was helpful for you, and well, I guess I'll be hanging around for some time for questions.
Yes, thank you, David. That was great in terms of getting a deep dive into all that you have been working on and really helping explain the biology and science behind mite management. Just asking people to, if they have further questions for David, to type them into the chat. And then there were a few slides that, Miranda, are you going to bring them up? Well, while we're waiting,
Yes.
some of them got answered in the chat, but while we're waiting for those slides to come up, I'll ask questions, David. One was just simply, are there opportunities to feed the orchard?
Hey, man.
Hey, guys, can you mute your phone? Mute yourselves.
Meeting.
Okay.
Is there opportunity to buy any beneficials of the beneficials in insectaries?
So great. So that's a great question. So first of all, I'm going to answer that question regarding predatory mites.
Yeah.
So we did a lot of work 15 years ago on predatory mites. A lot of releases of predatory mites. In fact, a major grower in Kern County had their own insectary, and there were thousands of acres were getting predatory mite releases. So if you were to ask me 15 years ago, I would say absolutely I recommend that all growers release predatory mites, because back in the day we were using Vendex and Nexter and Omite and all these miticides that we weren't using anything. The miticides being used are not used anymore, and all the miticides being used today didn't exist at the time. What's happened now is that six-spotted thrips is just taken over and it eats the predatory mites. And so I'm a huge advocate for insectaries, but I would hate for someone to go release 10,000 million whatever predatory mites in an orchard and have the six-spotted thrips just come and eat them all. So I don't really recommend that in the almonds anymore. For growers that do walnuts, for some reason, six-spotted thrips just isn't as prevalent in the walnuts as it is in the almonds. So maybe maybe consider releases there. As far as six-spotted thrips releases, I know there's places you can purchase them. I know some people that said they've released them, including in organic blocks, but there's no way to know what's, you put a card out and you catch the thrips. There's no way to know if what's being caught after those releases is what was actually released or if it's what was already there. And the fields we're in just have so many already that my gut hunch is that, my gut hunch is that if you buy them and release them, it's probably going to be in the category of a drop in the bucket compared to what's already out there. But if you want to do it and try it, go for it, encourage it. But there's absolutely no guidelines on release rates. Do you need 10 per acre? Do you need 1,000 per acre? What do they cost? What insectaries even have them? I actually don't know that information. I've just had a few growers say, hey, I bought some and released some, and there were a whole bunch, but I don't know if it's what would have been there anyways, or if it's what I released. So I don't know. So be a pioneer, go for it. But I don't have a scientific answer there.
I'm going to give you two questions, but then speak, but you can think about your answer. So the other two questions that come up is basically what density should the yellow sticky traps be placed in the orchard? And I'm going to add to it as to what are things to think about in the placement of those sticky traps. And the other question was, what impact does oil have the beneficials? But before you answer, I just wanted to remind growers that the Almond Board has put out some new resources, some of them with the help of David and so forth. But we're reissuing the UC IPM pest identification cards, and we've added the natural enemy identification cards. We also have some information related to irrigation management, some simple things that you can have in your truck, and then quick tips for spray drift application. This comes back to what David was talking about in terms of spray efficacy. Often spray drift is the inverse of spray efficacy. So if you can focus on spray efficacy, you typically will reduce your spray drift. Just to let you know, those resources are available. You can reach out to fieldoutreach@almondboard.com, or the outreach team has been planning to do what we're calling a road show. Once it's legal to do so, keep your eyes and ears open for when they will be in your area and show up, and they will be handing those this information out. Next slide, Miranda. So this is an example of one of those ID cards. And again, this is based on UC IPM's work, so we're just reissuing it. This is the example of the irrigation one. Next slide. Spray drift one. Next slide. And then again, as David was saying, is through a process at the Almond Board with the board of directors, really trying to encourage the increased adoption of environmentally friendly pest management by 2025. And I think really, David and I had the fun, I don't know how to jointly being on this chlorpyrifos advisory task force to DPR and to CDFA last year, and I'm now on the new advisory committee. I cannot emphasize enough how much California is wanting to change, wants to see us on a pathway to reducing the use of quote unquote toxic pesticides. And they're not talking about 20, 30 years from now, they're talking about as soon as possible. And we're seeing the same thing in Europe. So I just want to say, it's not just us saying this, we're only responding to what we're seeing out in the environment around us and trying to make sure that the growers have the tools they need to be able to grow almonds in this particular environment. Next slide. That's the codes, and I'll let, but so David, coming back to placement of sticky cards.
Yeah, so first of all, 2 per acre. 2 to 4 is what the research showed is adequate. I tell people just put one wherever a navel orangeworm trap is. Or sorry, did I say 2 per acre? 2 per field, if I said acre. Yes, not 2 per acre. Yeah, 2 per field. If you got a 40-acre field with 2 navel orangeworm traps, put 1 at each. And kind of the rest of this, Miranda, can you bring up the video?
And use that information in your spider mite management programs in almonds.
Were you guys able to hear that?
Yes.
Yeah.
Okay, great.
Feeding damage from web spinning spider mites can cause leaves to stipple, yellow, and eventually defoliate. However, almond trees are very resilient to damage, and yield losses only occur after severe tree defoliation. In these cases, losses are seen the year after damage occurs. When making spider mite management decisions, consider the number of spider mites and the predators present, as well as information like the timing of harvest, tree stress, the presence of dust, upcoming weather, and logistics, such as irrigation scheduling or the opportunity to incorporate a miticide with a pesticide application for other pests, such as a hull split spray for navel orangeworm. Presence-absence sampling is the traditional method used to monitor for both web-spinning spider mites and their predators. Begin monitoring for spider mites in mid to late March by dividing the orchard into blocks that can be managed separately. Select at least 5 trees per block to sample. Walk around each tree and choose 15 leaves at random from both inside and outside the canopy. Use a hand lens to examine both sides of the leaf for the presence or absence of both spider mites and their predators. In California almond orchards, common spider mite predators include 6-spotted thrips, the spider mite destroyer beetle, the western predatory mite, minute pirate bugs, and lacewings. Record the number of leaves on each tree with spider mites, as well as the number of leaves with predators. A sampling form that contains treatment thresholds based on the results of this presence-absence sampling can be downloaded from the UCIPM website. Currently, the main spider mite predator in California almond orchards is 6-spotted thrips. 6-spotted thrips feed almost exclusively on spider mites, are good at navigating tight spaces like spider mite webbing, thrive in hot, dry climates, and can quadruple their numbers in 1 week. The 6-spotted thrips adult is a small, slender, pale yellow insect with fringed wings. Adults can be distinguished from other thrips by the presence of 3 dark spots on each wing cover. Our recent research shows that using yellow 3x5 sticky cards is a highly effective method for monitoring 6-spotted thrips. We recommend using at least 2 cards per orchard and placing them near navel orangeworm or peach twig borer traps for convenience. Use a binder clip and an uncoiled paper clip to hang the traps at a height of about 6 to 8 feet near the edge of the tree canopy. Begin monitoring for 6-spotted thrips with sticky traps in mid-April. Check the sticky traps weekly through August and record the number of thrips on each trap. The spider mite destroyer beetle may also be caught in the trap and can be recorded on your monitoring form. In April and May, there is no need to treat for spider mites unless at least 25% of the leaves have mites and there is less than 1 6-spotted thrips per trap. Beginning at hull split, 3 6-spotted thrips per trap per week suggests a break-even point where there is an even chance spider mite numbers will go up, down, or stay the same. If there are 6 6-spotted thrips per trap per week, mites are almost guaranteed to go down and no spray is needed. For more information, visit the Almond Pest Management Guidelines online at the University of California Statewide Integrated Pest Management Program.
All right. Great video, David. Thank you.
Just some other questions that came in was, what's the impact of oil? Was one on the beneficials again, and then the other one is what to think about in terms of spraying young?
Good afternoon, everyone, and welcome. I'm Cheryl Reynolds, and I'm with the UC statewide IPM program.
Miranda,
And
that's the
welcome
next
to
YouTube
today's
video.
UC Ag Expert Talk on California.
Perfect, thank you. YouTube has a way to promote itself in the next video on the stream. So yeah, there's actually a lot of really good videos on the UCIPM. UCIPM has its own YouTube page with a lot of good videos on it now, a lot of different topics. So yeah, so oils. So oils smother mites. Okay, mites have a soft body, and oils just they smother them. You got to get good coverage, but they're quite effective. As I said, 50% reduction in the number of mites with an oil application, and that usually lasts about 2 weeks to 3 weeks, and the mites come back. But that's important to know because sometimes you don't need to, a hurricane when a light breeze will take care of something. And so, especially if you're close to harvest and, let's pretend you're at hull split and you do something and you're a week from harvest and you're like, oh, I'm a little scared. My thrips aren't quite here. I see some, but I don't think it's enough. I'm panicking. An oil application is going to knock back half those mites, and you're going to change that predator-prey ratio. So, the beneficials that are left now have an easier way to clean them up. So yeah, we do recommend oils, very beneficial, and oils make miticides work better, pretty much across the board.
And the oils have less of an impact on the beneficials compared to the mites?
So they can kill predatory mites the same way that they kill spider mites.
Okay.
No impact on the insect predators. So the beetles, the thrips, the lacewings, oil doesn't touch them.
Okay. And the other question was, for young orchards, what's your advice?
Yeah, so for young orchards, my advice is actually the same, except that I think I would, instead of using that, well, let me think about what I'm going to say here. Okay, so first of all, one of the problems with young orchards is that the biological control is not well established. And so I would say don't use that 40% of leaves infested threshold. Use that 25% of leaves infested. So use that. I mean, monitor biocontrol, but stick with the 25. Maybe even take that number a little bit lower, just because a mature tree can tolerate a lot of defoliation. A 1-year-old tree can't tolerate any defoliation. Okay, so come back down. So maybe come down even a little lower on that. But the other reason I say it's different is the dynamics are different. Like nowadays, in a mature orchard with biocontrol, we just don't really see the mites until after hull split. Well, if you go to a baby orchard and say, oh, I probably won't see mites until hull split, you could lose all your leaves in June. So it's just, yeah, use the 25% threshold or maybe a little lower. Make sure you're monitoring every week. Don't trust that biocontrol is going to save you, and then treat as needed. The other thing with baby trees is dust. Sometimes those baby orchards get really, really, really dusty leaves, and sometimes just coming through with 150 gallons of water targeting the trees just to wash off the dust and the mites can actually work wonders, in addition to an actual miticide spray. So yeah, a little bit different there,
Okay.
but not that different.
And other question was just simply a reminder of which of the miticides have less impact on the beneficials.
So as far as miticides go, all of the miticides across the board are safe on insect predators. So the thrips, the beetles, the lacewings, the pirate bugs, all of them are safe on all of those, with the exception of agromech and six-spotted thrips. So you're pretty good. Usually when almond growers kill the beneficials, it's not because of their mite programs, it's because of their navel orangeworm programs.
Yeah.
So, or their leaf-footed bug or stink bug programs. So pyrethroids, of course, are a problem. And then And then delegate. So watch out for delegate or intrepid edge with 6-spotted thrips. But delegate's an excellent thrips product, so be careful there. In fact, one thing to point out is with intrepid, for example, in pistachios where there's no mite problems, I recommend intrepid edge. It's nice because that's basically intrepid plus delegate. If you're an almond grower that's trying to rely on 6-spotted thrips for biocontrol, I would actually stick with the intrepid by itself, not intrepid edge, because the added delegate in there has the potential to have a negative impact on your thrips. So I don't think the Dow people would argue too much. Those are both their products. So, but pistachios, intrepid edge, almonds, intrepid in your rotations. But yeah, other than the delegate and the pyrethroids, that's pretty much the only way to kill beneficials these days. Now that the organophosphates are pretty much gone.
Okay. And then the other question that came up, since you mentioned that link between abamectin and leaf-footed bug, a question came in. Could you go back over that?
Yeah. So let me give you a 2-minute leaf-footed bug talk. Okay. So leaf-footed bug, of course, very sporadic, but sometimes you got to treat for it. So if you do, it's normally going to be around April, sometimes as early as March, sometimes in May, but it's going to be April-ish. And what do you spray with? So the most effective products are pyrethroids. There's no doubt. Brigade, Warrior, probably the top two. Both of those products are going to give you, they're going to kill everything that's there, plus everything that would come in the next couple of weeks. So if you still think there's more migration coming, that's your product of choice. Now, what impact did those two products have on mites? They just don't flare mite. Warrior flares mites more than Brigade, but anyone on this call that's an old cotton grower will remember that they used to use Capture on cotton all the time for mites. It captures Brigade. So Brigade itself has some miticidal activity. But so that's your main option. Now, do you always need to use a pyrethroid? No. So there are some other options out there. So belay can give you some knockdown of the adults. It's not going to have any residual activity. Likewise, Agrimec. Whenever we try Agrimec, it kills the adults that are in the orchard. But literally, if you spray it on the orchard, let the residue dry, and then cage a whole bunch of leaf-footed bugs on the dry residue, 1 hour later, it doesn't kill them. Okay, so and this was found out kind of accidentally, where we had a really, really bad leaf-footed bug year down in Kern County, and all the growers that put abamectin on for mites didn't have leaf-footed bug problems, and all the growers that didn't put it on did have problems. Even the same grower, 40 acres, yes, 40 acres, no. So we did some follow-up research to show that that's the case. So yes, abamectin is a leaf-footed bug product, but again, remember that if you spray it and a whole nother round of leaf-footed bugs fly in from wherever they're flying in, you're totally unprotected compared to the pyrethroid that's going to have some residual. So case by case.
So we are over time, and for those who want DPR credits, they still need to do the quiz. So I just want to
yes, do it.
Go ahead and do that. There was one more question that came in. It's more like, is there a way to spot treat for mites? Could you use soap and water and a hand sprayer?
Well, so sure you can. It's going to be hard to get to the leaves on the top of the tree. But so spot. So, okay, that was a little bit of a joke. Let me give you the serious answer. Spot treating, no. Edge treatments, yes. Okay, that's kind of a form of spot treatments. So it's common for mites to be the worst on the edges of the orchard, a little more water stress, a little more dust. Absolutely, it's totally appropriate just to mix up a load, spray the mites on the 5 outside rows, and oftentimes that's adequate to take care of an orchard. So yeah, maybe not spot spraying, but yeah, definitely the borders. And that could be with just an oil, or that could be with an actual miticide. Obviously, case by case, you have to decide what's best. But do monitor the whole orchard. I've seen cotton fields where growers do that, where they say, oh, look, there's mites on the edge, and they come in with an airplane and they spray all the edges. And the cotton field ends up looking like a picture frame where all 4 edges are green and beautiful and everything in the whole middle is black and dead. So, there's a time and a place for it, but, treat the whole orchard if you have to.